Welcome everyone. My name is Paulin. I'm a postdoc at Sedlazeck Lab in the Human Genome Sequencing Centre at Baylor College of Medicine. And today I will talk about Sniffles 2 and how it enables population structural variation, comprehensive population variation detection from Mosaic to population level. Let's start. Over the past decade, the role and impact of structural variants have become more and more relevant. Structural variants are loosely defined as 50 base pair or larger genomic alterations that fall into 5 categories: deletions, insertions, duplications, inversions, and translocations. And in some cases, complex events that are a combination of 2 or more of these types. Here we can see examples of each of them. For example, in the case of deletion, our sequence will have a missing part that is present in the reference. On the other side, for insertions, our sequence will have additional information that is missing in the reference. For the case of duplications, a portion of the sequence will be duplicated either in tandem, so that means next to each other, or they will have some other unique sequence in between for the case of interspecies duplications. For the case of inversions, the sequence is not changed, but some part will be in an inverted orientation when we compare it to the reference. And finally, for translocation, part of the sequence will map to a portion or one chromosome of the reference, while another part will map to another chromosome of the reference. Now, despite that single nucleus polymorphisms are in the range of 4 to 5 million per human genome, the structural variants impact the most number of nucleotides in the genome and account for the greatest cause of diversity given their size. In recent years, there has been an increased evidence about their importance across all categories of life. For example, they have important roles in evolution, causing speciation events. Structural variants also have a great impact in human disease, from Mendelian disorders, where deletions or duplications of genic regions have association with multiple syndromes. Also, in neurological diseases, where repeat expansions are associated with Parkinson's and Huntington's disease. And for the case of cancer, duplications of oncogenes, which lead to overexpression, are associated with breast cancer. These can be very complex, as we see in this example of a breast cancer cell line. Also, and connected with the previous example, they have an impact on gene expression, where they can lead to the disruption of gene function or a modification of gene dosage. Currently, our group is working alongside the GTEx consortium to further unveil the impact of structural variants in gene regulation. Finally, they have an impact on phenotypes across humans and all organisms, where including structural variants as a source of variation helps to better explain heritability of certain traits. Despite the importance of structural variants, these have been largely understudied because single nucleotide variation is more easy to discover. And given that, our understanding of how we study structural variants has been driven by technological advancements. But from the 1940s and 1980s, cytogenetics and karyotyping was the main way to detect structural variation. Here we could only detect large variations such as chromosome duplications or chromosome arm gains and losses. Then, with fluorescence technology, we were able to have a better resolution on these translocation events. Next, following, microarray data gave us access to gene gains and losses. Finally, with short and long-read sequencing, we're now able to detect more structural variants. However, Illumina sequencing is not able to easily identify these events, as it detects roughly between 10 to 12,000 structural variants per human genome. On the other hand, with long-read technology, we're able to detect twice the number of structural variants per genome. But not only the number is important, as long-read technology gives us access to difficult genomic regions, as we're able to detect previously inaccessible structural variants in medical relevant genes that cannot be accessed with short-read data. Highly repetitive sequences, which also include the centromere and the telomere. With both Oxford Nanopore and PacBio improving their sequence quality, and soon reaching levels of Illumina-like errors along the genome, long-read technology is no longer a novelty, and population and production scale studies are now a reality. We can see here that PacBio HiFi has improved accuracy with the drawback of the reads not being too long, with Oxford Nanopore falling behind in accuracy, but reaching read lengths that in the hundreds of kb with their ultra-long technology. Here we can show you in a red line what Illumina advertised as the maximum error rate, and we can see that PacBio has already achieved Illumina-like error rates, while ONT is still falling behind. But how do we detect structural variants with long-read technology? So here are 5 examples, one for each of the structural variant types. So for deletions, we will have a split read, meaning that our initial sequence we will split in two because there's a missing part that is present in the reference that is not present in our sample. Next, for duplications, we will have increased coverage as different portions of a given read can be aligned to the same sequence. Next, for inversions, if the read is long enough, we'll see part of it is in inverted orientation compared to the other two. Next, for insertions, we also get a split read, but in this case, a part of it will not map to the reference as the sequence is missing. Next, for translocations, one part of the read will be mapping to a specific chromosome, while the other part of the read will be to another specific chromosome. Given the importance of structural variants, we're still struggling to detect germline or even robustly identify de novo structural variants. So it is here that we introduce Sniffles 2, our redesign of the popular SV color Sniffles. So first, Sniffles 2 improves on germline SV calling, being more accurate and faster than currently popular methods. But we didn't stop there, and we enabled new applications in the realm of structural variant calling. First, by solving the problem of population-scale structural variants with long-read technology. And second, by enabling the detection of low-frequency structural variants across datasets. These low-frequency SVs are also called as non-germline, somatic, or mosaic SVs. So first, in order to test Sniffles 2 improvement on germline calling, we make use of the two genome in a bottle benchmark datasets. These datasets are structural variant truth datasets that were compiled by manual curation using multiple sequencing technologies. We use these benchmark datasets to first assess the performance of Sniffles 2, and second, to compare Sniffles 2 to other available tools, including Sniffles 1. The first dataset we use is a genome-wide benchmark for the identification of large insertions and deletions. The second one is a characterization of challenging medical relevant genes that are not accessible for short-read data because they are too complex, include highly repetitive sequences, and segmental duplications. As a metric, we're going to use the F1 score, which combines precision and recall in a single metric. Here, we're using the genotype F1 score, which not only takes into account the presence or absence of the structural variant, but also its genotype. We can show you here, with the blue arrow, the performance of Sniffles 2 in both F1 genotype and CPU time. And as you can see, the Sniffles 2 has the highest performance with compared to all methods that are here shown, which are Sniffles 1, CuteSV, PBSVIM, and the SVIM. So first, we can show you that we made the comparisons with different coverages, from 50 to 5x. And one thing that goes first is that at low coverages, many of these cannot even compete with Sniffles 2. This is because for Sniffles 2, we have an automated parameter selection for filtering structural variant candidates based on available coverage. In contrast, for other SV callers, they rely on manual adjustments of these parameters to retrieve acceptable results across coverages, and in some cases, even for sequencing technology. So what we did next is to give them a chance to compete with Sniffles 2, is we optimize the parameters for these other colors. As we have shown that with default parameters, Sniffles 2 strongly outperforms them at low and medium coverages. But then, with optimized parameters, we give them a chance to improve their both accuracy and speed. As you can see on the right-hand side, all tools had improved accuracy when we optimize these parameters, but none of them come close to Sniffles 2, which remains the most accurate and fastest of them all. Next, we go to the challenging medical dreams. In this case, the results, as you can see, for in both the F1 genotype and speed, Sniffles 2 is better than all other available tools. When we compare the accuracy and speed of Sniffles 2 against the other tools, we can see that if we take the second best, which in this case is CuteSV, for these results, CuteSV took almost 10 times the time in CPU time. On the other hand, if we take the second fastest one, which is SVIM, the result of Sniffles 2 is 12% more accurate. And thus, we show that Sniffles 2 is more accurate and also the fastest of all current tools available for structure variant calling. So next, we have shown you first now that Sniffles 2 is now the best in general SV calling. So now we go for a different approach, and we perform new applications in the realm of structural variant calling. In this case, a recent review by our group has shown that the number of individual sequence with long-read technology is rapidly growing. Just in the past 3 years, we went from 10, 12, like in the tens numbers of structural variant, individuals sequence with long-read technology to the thousands. And future studies aim to reach 1 million individuals sequenced with Oxford Nanopore technology. For this reason, we have implemented in Sniffles 2 a population mode, which will allow us to have a full SV genotype from family trio to populations. Current pipelines are slow as they require multiple steps. Here we show you an example with CuteSV. So the first step for both CuteSV and Sniffles 2 is to call structural variants for each of the individuals in the population. In this case, for this example, we have a family trio: mother, father, and province. The second step for CuteSV will be a merge of the structural variants that were called for these three individuals. Next, we will do a regenotyping step, which will require to read the alignment files again. This step is very slow. After this slow step, we'll do another merge in order to get a fully genotyped population VCF file. In contrast, for Sniffles 2, there's a single merging step, as we have created a special binary file that contains structural variant candidates, which are then used for the merging step without the need of reading the alignment file again. Avoiding this significantly decreases the runtime, and thus we're able to produce a fully genotyped VCF file much faster. For this same example, Sniffles 2 took only 65 CPU seconds, which means that we are 2,000 times faster than current methods. But not only that, Sniffles 2 also solves the N+1 problem, as we can add new samples very easily. With previous pipelines, we will need to perform all the merging, regenotyping, and remerging steps for each sample that is added, but not for the case of Sniffles 2. We just need to add the new samples to the merging step. Finally, we will show you that we can scale up this to a population level by performing a merge of over 1,000 samples. So first, we assess Sniffles 2 population merge using the HG002 family trio by calling structural variants from the mother, father, and province, and then performing the merge in order to obtain the fully genotyped VCF file. Here, what we measure are the Mendelian error, meaning consistent genotypes are those that can be explained by Mendelian inheritance. Those that are inconsistent are cases that violate Mendelian inheritance, and those that are missing are cases in which the tool could not assess the genotype of a specific structural variant. We can show you here with the blue arrow that Sniffles 2 has a high number of Mendelian concordant genotypes, a low number of non-concordant genotypes, and a negligible number of incomplete genotypes. When we compare these results to CuteSV and a simple merge in the middle, we can see that there is an increase in the number of incomplete genotypes. Then, if we perform the forced call and the regenotyping and remerging, we can observe that these results improve. However, not at the point of Sniffles 2, as these results from CuteSV have a lower number of concordant genotypes, a higher number of non-concordant genotypes, and a higher number of missing genotypes. For stress test, we performed a merging of 1,002 genomes, which means we had 334 family trios. This endeavor took Sniffles 2 9.25 CPU hours, which means that we are 4 times faster at merging 1,000 genomes than it is for CuteSV to perform a family trio. So next we apply the population merge of Sniffles 2 to a dataset of 31 samples that contains 7 full families, meaning mother, father, and province, with known disease status. Sniffles 2 population merge was able to detect structural variants that are disease relevant that affect the MECP2 gene locus. These lead to neurological and developmental disorders, primarily in males, as many of these structural variants are inherited from the mother. In this first example that we show you, it's a duplication that's coupled with an inversion. So on the left-hand side, it's a first of the 2 duplications that we can see, and that one affected the MECP2 gene locus. The second one on the right-hand side, it's near the MECP2 gene locus, and it's also a duplication. When we apply Sniffles 2, we can observe that there were also 2 inversions that affected both the duplications. And in the last part beneath, we show you the reads that connect these inversions that were detected by Sniffles 2. So next, we'll show you an even more difficult structural variant to call. This is a very complex event that combines multiple of the structural variants. So the first one, A, it's a duplication. This was obtained with array data. Second, in B, it's a triplication. And the third one in C, it's again another duplication. So the second track that you can see is the array data, in which the black dots means that it's like regular coverage. Then the log ratio goes to 1 because it means that double the alleles here. So we have a duplication in A. And you can see there's missing part between the duplication and the duplication. This is because there's a segmental duplication here, marked in gold. And this here, the array cannot assess data. Next, with Sniffles 2, we're able to detect an inversion in the first duplication, shown here in green. And all of these affected MECP2 gene locus. Beneath, we show you the split reads that we used to identify this inversion. Those are colored in pink and blue. Finally, you can see then that many there are kind of gray reads in beneath, and there are some that are white. Those are showing the segmental duplications. So in this case, Sniffles 2 was only able to detect one of the 2 junctions because the other one falls within the segmental duplication, and thus it's very hard to detect them. So we can show you here that Sniffles 2 is a great aid in order to detect these very complex events. In the last part, we can show you how this is linearized, meaning how this is all connected in the genome. As you can see, these very complex events not only need one single technology, but multiple ones. And here we can show you that Sniffles 2 is able to aid greatly in the detection of structural variants. So finally, for another application, we turn to low frequency structural variants. And so we have implemented in Sniffles 2 a non-germline mode. Multiple studies have shown that low frequency structural variants, also known as somatic or Mosaic SVs, are likely causal across neurological disorders and oral diseases such as cancer. But how does this work? So first, here we show you a benchmark dataset. This dataset is 70x coverage and contains mostly reads from the mother. So we have here created this benchmark dataset with reads from the mother and a son. Most of the reads come from the mother, and we have spike in various concentrations of the son. Both individuals share genetic background, however, there will be differences. And these differences will be some structural variants. Here you can see most of the reads do not show any signal of the structural variant, but 3 of the reads shown in purple. These 3 are insertions that were called by Sniffles 2. And when we compare the results of Sniffles 2 to the genome in a bottle gold standard, we can see that these insertions are actually true positives. So next, if we apply the default mode of Sniffles 2, we cannot observe anything. So here we show you the titrations of these reads from the mother and the son, which are 65x for the mother and 5x from the son in the first sample. Next, 63 to 7, 60 to 10, and 55 to 15. We can show you here in the first dot, for example, in the 65 to 5. For this case scenario, many of the reads that contain the structural variant are in the range of 1 or 0, meaning that it's very hard to detect them with a single read. Still, we were able to detect 35% of them. Just increasing a bit the sequencing depth of this spike in events, we can see a huge increase in the recall. And this is done without affecting the precision of Sniffles 2. The gray line will represent here it's a force call, meaning that we give Sniffles 2 the information where the structural variants are present, so it just needs to call them. When we compare the final dot on the top, which means that we have 20% roughly information from these low frequency structural variants, the result is almost comparable to a force call. So in the gray line, the information is already known, but in the orange line, the information is not known. And thus showing how good is Sniffles 2 at locating these low frequency structural variants. So we apply this to a long read dataset of 50x coverage of original brain sample. This sample is from an individual that has a rare neurodegenerative disorder called MSA. This disorder has progressive autonomic dysfunction and Parkinson-like symptoms. Here we show you on the right side the actual data. This is not an example. So here, most of these reads do not contain any information regarding the presence of a structural variant for 3 of them. Here, Sniffles 2 with the non-germline mode was able to capture a rare structural variant that was missed both by Illumina at 90x coverage and by optical mapping with BioNano at roughly 700x coverage. You can see here that this is a deletion, and this deletion is in a repetitive sequence in a gene that is important for MSA disease progression, and thus showing that Sniffles 2 is able to recover these rare events that are disease relevant. In summary, we have shown you that Sniffles 2 has increased accuracy across established benchmarks, and it strongly decreases the computational cost as it is the fastest of the available tools. Then, we presented you new applications. First, population scale structural variant calling. And we applied these to a dataset and detected the role of structural variants affecting the MECP2 gene locus in Mendelian disorders. Then, with the detection of low frequency structural variants, we show you that Sniffles 2 is able to detect somatic SVs in neurodegenerative disorders. Sniffles 2 is available as a contact package, and the preprint was released in early April. Now, I would like to thank everyone involved in this project. First, the Sedlazeck lab, especially Moritz Moka, who is the main developer of Sniffles 2, and also all collaborators, Claudia Carvalho from the PNRI, and Christos Proukakis from the UCL, and you for your attention. Thank you.